And with that, I want to introduce our guest expert, Rebecca Smith. She is a clinical fellow here at Vanderbilt University Medical Center. Rebecca received her PhD from the University of Alabama at Birmingham. She completed a postdoctoral fellowship at St. Jude's studying molecular mechanisms of ALS. She has about 20 years of experience in biomedical research and 7 years in clinical laboratory diagnostics. As I said, she is currently the ABMGG Laboratory Genetics and Genomics Fellow, which means that there is a combination of molecular and cytogenetics training involved in that here at VUMC. And she will begin a new role here in June next month as a laboratory director at a Nashville-based cytogenetic and molecular pathology testing company called Genetics Associates. Congratulations, Rebecca, on your new job.
Thank you so much.
Yeah, and with that, we're very thankful that you're here presenting for us. So just let me know when you need me to navigate for you.
Okay, thank you. Well, I really appreciate you inviting me to talk today. I'm really passionate about this topic, about variant classification and NGS reporting, variant reporting, so that the clinic can have a better understanding of what's being reported and how they can use that information. So, yeah, I guess the next slide. So this is just the talk outline today, and just to kind of warn you, I do have quite a few slides, so I'm going to try to just really talk about the main topic in respect of the time that I have today to kind of talk about the highlight of each slide. So for the topics that we're going to be discussing, really the information that is included in a report, NGS report that you might see in the clinic. And then we're going to talk about how the variant was classified and if there's any investigation that the clinic can do on the pathogenicity of the variant. So we can go to the next slide. So really there's critical information that should be included in all reports. And this comes from the CAP molecular checklist published in 2019. And I have seen some NGS reports that don't have all of this information. So, you know, it makes it frustrating, I think, to see these reports where you don't have enough information to be able to look up the variant and find some more information, or even, you know, accurately give the patient the full information about the variant. But some critical information, according to CAP, would be the gene name, genomic coordinate, like chromosome 13, and then the coordinate on the chromosome, the transcript reference number, coding information, and this is the information about the coding transcript, the nucleotides, but it's excluding the introns. So I wanted to just briefly talk about this. So in this case, this is a coding location of 369 through 372. It has a deletion, and there's a deletion of 4 base pairs. And this would indicate that this change is going to, since it's not a replicate of 3, it's going to change the frame, the reading frame of the nucleotides. Another variant that you might see in reports would be a coding nucleotide location with a plus or minus. In this case, like the coding location could end, let's say exon 4 would end at 506, and exon 5 would pick up at 507. But when you have the plus or minus, this is indicating that this is in the intron of the variant. And this is important. Many testing doesn't include deep intronic variants, but variants that are right at the end of an exon are canonical splice sites. So if you see a +1 or +2, you know that that variant will impact the canonical splice site of that gene. Also, the reference genome is an important aspect to consider in these reports. So most companies use version 37, which is kind of an older version, but I have heard that some companies are starting to use 38. This is important because many companies, their databases are based on 37, so that's why there's a reluctance to shift to 38. But if you find a report that has kind of a differing nucleotide location, you would want to look to see, are they using 37 or 38? Because it might be just the case that the same variant change in genome 38 has a little bit different nomenclature. So it could be the same variant, just a different naming convention. Other critical information that wasn't directly mentioned that's required by CAP was the protein information, which has a P right here. An RSID, which is a reference SNP clustering ID, and the variant allele frequency. And I think the allele frequency is important when determining if something is inherited or somatic. So if you're looking at a somatic test, but you see a variant is around 50%, you might consider that, you might want to investigate that variant for germline inheritance. And a variant allele frequency of around 50% would indicate that it is inherited as a heterozygote, so it has one copy. But if you see something at 100%, you might consider, well, we might have a inheritance pattern of a homozygote, 2 copies of the same variant, or the other chromosome could have loss of heterozygosity, so it could be lost either by a deletion or just the same chromosome being duplicated there. Oh, can we go to the next slide? So I wanted to kind of talk about a case that I saw when I was rotating in our clinic. And in this case, there was really not enough variant information in this report. This was a 61-year-old male with a diagnosis of prostate cancer. And there was in the report for the somatic testing, there was a BRCA2 positive variant. And the question from our clinic was, could this mutation have been inherited? And the patient was interested in the implications for inherited variants for his children. So an investigation with an Invitae inherited cancer panel was employed, but Invitae needed to know what was the prior reported variant. Well, unfortunately, the report only included this information, the protein information. It wasn't even complete protein information and no other information. And it could have mapped back to a variety of different nucleotide changes. So if you could go to the next slide. So it was kind of frustrating at that point that we couldn't determine the patient's true nucleotide change to actually tell the patient or another testing company. If you could go to the next slide. So we did get more information in this case, and we were able, with the transcript ID and the coding location, we looked up the protein, the change, and we also found that the variant allele frequency was around 77%. And I think that's important because, it gives the clinic more information. In this case, this was in a somatic sample, whether they would want to investigate whether this variant was inherited or not. Was it around 50%? And so this one might be questionable in this case. So the clinic might have decided not to investigate it further, but it really gives the clinic the most information if they have the variant allele frequency. So you can go to the next slide. And so why is this information important? And I'm going to start from the right side here. So when we just have the protein information, we're really just getting, it's sort of analogous to trying to find your friend's house that you've never visited before. So you have to really have the address, the complete address, in order to find the house correctly. So if we start on the right, we're really looking at the protein information. So this is giving us information about, what does the house look like? If you just had, well, I live in a yellow house with a red door, you're not going to be able to find that house. But if you have the coding location, similarly, you can, it is analogous to maybe the street address. Well, I live at 100 Meadow Drive. Well, that's not going to help you get to that variant unless you have the city and, the state. That would be very helpful. So you really need this transcript ID when you're looking at coding location in order to unambiguously map back the variant. And the genomic coordinates are very helpful because you can get to the variant change. It's sort of analogous to a longitude and latitude in that case. So you can find the exact location. It's not giving you information about whether there's a gene present, but it's really helpful to have this in the report in order to make sure you're at the right location. You can always get there with the genomic chromosome and coordinate. So if you could go to the next slide. And I wanted to talk briefly about the transcript ID, which I think poses a lot of problems in the field right now, and I would really hope that it could be standardized in the future. So this transcript ID is called a RefSeq number, and it is from a project at NCBI, and it's an available database of annotated genomic, transcript, and protein sequence records. So the main take-home message of this slide is that transcripts typically use this NM prefix number, and that corresponds to the mRNA of the gene. The NR number is non-coding RNA, and NP is the protein. So whenever you see a coding number, it should have a associated NM a transcript number. And the difference between these prefixes and maybe something like XM, XR, XP is that these RefSeq transcripts are known transcripts, but the XM, XR, and XP are only predicted. So they're not, in fact, actually established true transcripts. So really, most all places that I have really seen use the NM number. That's the most important one. So if you could go to the next slide. So I wanted to kind of show you how to look up different NM transcript numbers. And what does this really correspond to? Well, different transcript numbers for a gene would correlate to different splicing isoforms. And I kind of showed you the steps how I looked this up. At the NCBI website, there was a drop down where I selected gene and I typed in NF1. And if you see this gene comes up and it has underneath RefSeq transcripts 3. So I can click on that, and then some more information pops up that you can see to the right. It lists 3 different transcripts of different nucleotide links. And the last one is quite a bit different. And there's a map at the bottom of this information page that has the different transcripts. So you can actually visualize what the different transcripts are indicating. So you can imagine that if you have a variant change, it depends on which transcript you use to what the variant coding information is going to be. And I've read that typically people should use, or companies should use, the longest transcript. Maybe that's in order to have the most information, but I think many companies just use the transcript that's labeled number 1. So that's something to keep in mind when you're looking at reports, to understand the transcript that is being used. So if you can go to the next slide. So this is a case that I encountered in the clinic during my rotation, and it did present a problem. And this is just an example for how to look up different transcripts. So we had a case where we had somatic testing and then germline testing. And in these cases, we had 2 different variants. In the first report, we had one with a protein location of codon 2201. And in the other report, we had a protein location in codon 2222. So our question was, are these the same variant or different variants in the NF1 gene? Are these different mutations that are occurring in NF1? But luckily, one report listed the RSID, and we could do a search at dbSNP. And I listed this link just so you could follow along at it after the presentation. So if you could go to the next slide. So when I want to verify the transcript ID, I usually visit the website dbSNP, because this has a really nice list of aliases here. So if you can see for this variant that we looked up, you can see its coding location at different transcripts. So in transcript 1, it has a coding location of 6664. And in transcript 2, it has a coding location of 6601. And it also lists the protein location, which we saw on our report. So in this case, if you're ever, challenged, like, is this the same variant? Maybe they're using a different transcript. You can check it on dbSNP. And if you go to the next slide. So from our investigation into dbSNP, we found that these two variants are the same variant. They're just reported using a different isoform transcript. So we were able to conclude that they were the same, and we didn't have to stress the family out further with that information. If you could go to the next slide. And then one more. Great. Well, so now we've talked about some things that you'll see in the report. And I think it's very important for unambiguous mapping of the variant. I really hope that there's some standardization of reports in the field. I think there's definitely movements towards that. And that would be great for even literature publications and reports in order to reconcile their information. But I also wanted to talk briefly about how the clinic might investigate pathogenicity of the variants. And this is just from some experience that I have as a clinical variant scientist in industry. So could you go to the next slide? So inherited variants are classified based on the ACMG 2015 guidelines. And they have 5 categories of classification, going from benign, likely benign, variant of uncertain significance, likely pathogenic, and pathogenic. So when these variants are being classified, we're really acting as detectives. And we're trying to find evidence in databases and literature to build a case against the variant. So, and different evidence has different strengths in the classification. So I just mentioned a few. If there's a functional study, it's considered strong evidence. If there's other known patients with the same variant, it could be strong or moderate. Other information could include if the variant is in a critical hotspot, if it matches other typical disease-causing mutations in that gene. Are they all truncation variants, or are there missense variants? And something we'll talk about more are in silico prediction, and is the variant in control databases as a rare variant. So in order to classify, if we kind of look to the right here, in order to classify variants as likely pathogenic or pathogenic, we have to have certain combinations of the evidence. And if you note, in most cases, you'll have to have at least one strong piece of evidence. And I'll, if there's time, I can touch on that a little bit later. So, if there is a variant that you come across that's not in any databases, that's never been described before, it's going to be really hard to get it into the likely pathogenic range because it doesn't have any evidence that would point it towards likely pathogenic. Okay, could you go to the next slide? So when you encounter a variant, there are 3 really low-hanging fruit that we all can assess when we encounter that. And this could help for consultation with the family. I know that some of the reference labs are doing the classification, and really, they have quite a bit of experience. So this is just kind of building on the information they provide. So we can assess whether the variant produces a premature stop codon or alter a canonical splice site. We can also assess if the variant is rare in controls. So we can look in control databases and see what the population frequency is in those controls. Also, we can assess the in silico prediction model of pathogenicity. So if you could go to the next slide, I can walk you through. This is a very busy slide, and I'll just kind of touch on it briefly, but it has the 3 points that I just spoke about. Really, one of the easiest variants to classify would be one that produces a premature stop codon or an altered canonical splicing site. And this evidence would be very strong evidence, almost standalone evidence that this variant is pathogenic. But there's some caveats. The gene would have to be, its mechanism of disease would have to be loss of function. And this premature stop codon would not be in the last exon or 50 base pairs within the next to last exon. And this is because premature stop codons earlier in the transcript would produce nonsense-mediated decay of that transcript. So really, it never makes it to become a protein. Of course, there is leakiness in nonsense-mediated decay. So even if there is a earlier stop codon, it's likely that some truncated product is being made. But that's why, we don't know if the premature stop codon is in the last exon, how the effect of the transcript is. It could still be, a functional transcript in protein. Next, we can, for number 2, examine the allele frequency in the population databases. And the main take-home points here are that if you find your variant in a population database, that means it's kind of in a control population. It doesn't necessarily mean that all those controls are healthy. It's healthy to a certain extent, but maybe they haven't had a disease onset yet. But if you see a population frequency of above 5%, which goes to this purple square, that's automatically benign evidence. So you can classify that variant as benign. If it's less than 0.01, that could be considered a rare variant, and it would be moderate pathogenic evidence. And then lastly, we can reference in silico models of computational pathogenicity assessment. So it's kind of a prediction. So could you go to the next slide? So I wanted to just briefly show you a really helpful website that I use often when I'm kind of trying to see what the protein effect is. And so this is called Mutilizer, NL, and I think it's really helpful to investigate premature truncation variants, or really any variants if you want to kind of get a good idea of what's happening in the protein. So you have to type in the transcript number and the coding information, and it'll tell you what the protein change is using correct nomenclature. So that's also really nice if you ever want to verify the protein information. And it'll also give you information on the exon. So in this case, I know the font is really small, so I can't see. I think it's like 369 to 367. We can determine that this is an exon 4. So we know that although this is going to be a frame shift, it's an exon 4. And so that's not within the, if it was within the last exon, we might be a little more cautious in calling this a pathogenic variant. So if you go to the next slide, I can show you kind of how this is really nice on the display. So this is a variant that I typed in, and you can do this exercise later if you want to get comfortable with the website. And you can see this is the reference protein. This is for TP53, and this is the change that's produced by the variant. And you can also detect the exon that it's in. So it's within exon 7. So it can give you a picture of what's actually happening to the protein. Could you go to the next slide? So another question that we'd like to answer is if the variant is rare. So you can use the website Nomad to find population allele frequency. So in this case, you can see that the allele frequency just does show that it's very rare. And I think Nomad is the most preferred website right now to do this at. It's maybe the most up to date. But some other websites that you can use to cross-reference would be 1000 Genomes, Exome Variant Server, or DBSNP. So if you could go to the next slide. So another website that I'm using often is called Marvel. And this is the Model Organism Aggregated Resources for Rare Variant Exploration. And I think that this is really helpful because you can type in the variant change and a lot of information comes up. And it has a really nice prediction summary of all the different websites that you could check, and it shows the prediction score and the rank of this variant, and on a scale of 0 to 1 on how pathogenic it is. So if it's closer to 1, it's considered more damaging. So this is something that you could look up quickly, when you get a variant report in, and you can see the results from several different in silico prediction sites for that variant. But you could also use polyphen-2, SIFT, and amino acid conservation data. So that's some evidence indicating that the variant is pathogenic if the amino acid is well conserved. Okay, can you go to the next slide? I think I want to conclude, but the last thing I want to say is I was trying to think, we often send out NGS to reference labs, and I wonder if they have as much information as we have in the clinic in classifying these variants. So one piece of information that these reference labs don't have is the co-segregation of this variant with disease in the family. And this is a supporting evidence in ACMG guidelines. So I think that, if you have a variant that you're looking at that's in a family, you'd have to have several family members tested for that variant. But it is segregating with disease. It might be an opportunity to reach back out to the reference lab that's done the classification and say, well, this is the evidence that I have about co-segregation. Would this increase the pathogenetic score of this variant for this family? And it is supporting evidence. As I mentioned before, strong evidence is what really pushes it in one direction towards pathogenicity. But it might be, just an extra piece of information needed that could provide more support. So I think that's how I would kind of approach the situation when the reference lab doesn't have that information about the co-segregation within a family. Okay, you can go to the next slide. And I think I'm going to try to conclude there, because I did mention that, to achieve a pathogenetic score of likely pathogenic or pathogenic, you really need some strong evidence. I guess I can go through this very quickly and then I'll wrap it up. So the strong evidence includes whether this variant is at the same amino acid change as a previously established pathogenic variant. I think that makes sense because, if it's the same amino acid change, it would also cause disease. If the variant is de novo in a patient with a disease, and in that case, you'd have to do trio testing or to determine that variant is de novo. A functional study, a research study done on this certain variant showing that there's a decreased function is a strong piece of evidence. And if we can find powerful case control studies showing that the variant is increased in affected individuals. And the ACMG guidelines do specify that these have to be statistically significant. But I think I'm just going to wrap it up. If we can go to I think you can skip the next slide. Okay, here's the summary. So I really feel strongly that there's critical variant information that should be included in all reports, and it is mentioned as a CAP requirement. So I hope that becomes more standardized. We classify variants using evidence from databases and literature. And, to push a variant into the pathogenic or likely pathogenic range, we do need a strong piece of evidence in that case. So I think that's going to conclude my talk. And I'd like to thank you all, and I'd be happy to take questions.